Okay, welcome everybody. Glad to have you here for our scientific session. Today I'm going to be telling you about a recent initiative that we've begun on exploring the genetic diversity of our diversity outbred mice. So the diversity outbred mice are a colony that we have here at JAX that is specifically bred to have diverse genetics within the population. So it's a very different breeding scheme from our typical inbreds that we sell. And I'll go through a little bit of that as we go through the presentation. So this is just a description of how the diversity outbred population was created. It was created at first with combining 8 different founder strains. Now, each one of these founder strains are inbred mouse strains that were combined together through a very specific breeding mechanism to sort of mix all 8 of these inbred strains evenly. First, this mixing was done through the creation of the collaborative cross lines. If you've never heard of them, it's something very easy to Google. It's basically a series of recombinant inbred lines that have each one of these 8 founder strains within them, sort of mixed up in different patterns. Then once those collaborative cross lines were created, those collaborative cross lines were bred together in a random way and in a random way with each generation in order to create what we call the diversity outbred population. So the goal of this population is that no mouse is ever the same as any other mouse within that population. So much like the human condition, each mouse within that population and over the generations is unique and can never be found ever again. But each population of diversity outbred mice should have approximately a 1/8 contribution from each one of those 8 founder strains. So that's sort of theoretical, and what I'm going to show you today is what's actually going on in this population and whether or not it is close to what we believe is happening theoretically with their genetics. So this is a little bit more of a nitty-gritty of how we maintain this colony at the Jackson Lab. So essentially, for this entire population, we maintain them through 175 breeding pairs each generation. What happens once those 175 breeding pairs are produced is, I'm trying to pull up my laser pointer here. Once each one of those 175 lines produce offspring, is that a subset of those offspring will be set aside for mating in the next generation. All the progeny that are left over from that series of matings can be sold to customers. Then what happens when we get ready to mate the next generation is that each one of the females sort of stay within their line number. So we sort of track the lineage of this population through the maternal number that we ascribe to them. Then we randomly mix up which male she will be paired with in the next generation. And we do that so on and so forth. We have 4 generations occurring of this population every single year. And what we try to do with those leftover mice that we set aside for sales is that we mix those animals in boxes that we ship off to customers. So in any one box, you're going to have animals that are non-siblings, and we try to maximize those numbers of non-siblings per box as much as possible, so that essentially you're getting in a shipment as much genetic diversity as we possibly could give you within this population. Now, a lot of people use this population for sort of screening of quantitative trait loci, so how do genetics map onto phenotypes that you're interested in. We know of other people that have used it for toxicology or pharmacology, basically screening how therapeutics would work in a more diverse population with lots of alleles leading to different conditions and disease states within this population. If you're interested in learning more about the uses of this population, we have a technical information webinar on our website all about the JADO and how people have been using this population over the past several years. Because at this point, we are about generation 40 of this population, so we've been maintaining it for almost 10 years now. And that's one of the reasons why we did this analysis that I'm going to tell you about. So essentially what we wanted to do is that this population and its sort of founder contribution, so how much of each one of those inbred founders that sort of started off the population are contributing to the current population, has not been assessed since generation 21. There was a big paper that came out at that generation that assessed a whole bunch of genomic things that were going on with this population. And, you know, essentially we're at generation 40 now, so it's been almost 20 generations since we've looked at how the genetic diversity is going in this population and whether or not our breeding schemes have been maintaining it properly. So this whole effort was in conjunction with our computational sciences group here at the Jackson Lab to basically look at how our population is doing. Basically a quality control for our customers who are buying diversity outbred mice to assure them that the mice that they are receiving are diverse and that the founder contributions that they're expecting are actually there. So what you'll see in this slide is that in generation 21, which is what we expect to sort of be our gold standard, the last time that we checked on this population in terms of genomic contribution of our inbred founders, you can see the percent of each one of our founders represented by the different colors during generation 21. And so this is looking at the population as a whole. And what you'll see is that our theoretical is that each one of these founders should have a 12.5% contribution to the overall genomics of a population. And you can see that some of our founders have less and some have more. You can also look back at this scheme here just to sort of understand that the colors we use for each one of our founders. So red is PWK, which is one of our wild-derived strains, and you can see that's one of the ones that has a little less contribution to the overall population than some of the others. But what you'll see is that throughout the generation, so in this particular case, we had enough data to assess generation 22, 23, and then we didn't have enough data for a couple generations, but all the way from 29 to 34, where we had our most recent data for genomics, because a lot of people that use these mice keep them for a long time on the shelf. So even though we're breeding generation 40 right now, there's not genomic data for generation 40 at this point. So 34 is the most recent that we could get for this type of analysis. But what you'll see is when looking at the population as a whole across each one of these generations, the percent contribution from each one of our founders is really consistent across the generations. And we were so happy to see this, that we're not having big fluctuations in founder contribution, that we have maintained sort of what was there since generation 21 from our breeding scheme of this population. But we also wanted to look a little bit deeper. That's sort of the big picture. It's the whole population and looking at all of their genomics. So there's a few ways that we can break this down a little bit more. Instead of just looking at founder contributions for all of generation 21 across all of the chromosomes, we can break it down across the chromosomes and see founder contribution on chromosome 1 versus chromosome 2, and so on and so forth. So what you'll see here is that even though our averages are about what we expect, 12.5% across the whole entire genome, within generation 21 and all of the animals that we analyzed in that generation, there is variability across the chromosomes. This is not unexpected. That the mixture that we originally got from the collaborative cross lines being crossed together could possibly contribute to some of these variations across the chromosomes. But one thing I would like you to notice that this is once again the profile of generation 21, our sort of gold standard for analysis and genomic profile of this population. And what you'll see is that in generation 34, our most recent data, the sort of look of this profile is very similar. There are some variations, minor fluctuations in percent contribution across each one of these chromosomes within this generation. But all in all, it's very consistent. So once again, we were really happy to see that the breeding strategies that we've been using have been maintaining at least what we had since generation 21. We can also dive a little bit deeper into this data as well, where we can look at within an individual chromosome, so each one of these panels is the colors of the different founders within only chromosome 2 in generation 21. And what this shows you is sort of the spark plot of contribution across the length of that chromosome. So what you can see here is if you consider the beginning of this chromosome, so the far left on each one of these panels, you can see that there's a lot of contribution by 129S1 within that region, and less so of PWK and CAST in that sort of beginning region of chromosome 2. And you can see how those founder contributions change across the chromosome itself. So once again, we're diving deeper into sort of the nitty-gritty of that founder contribution within the chromosome. One thing that you may notice by looking at these spark plots is that in WSB, we have a region that's at completely 0 in generation 21. And what that is that was actually an effort that was made by us in generation 21 to eliminate an allele contributed by WSB that was leading to a meiotic drive. So essentially, there was a region in here that was allowing WSB to go to fixation within that region. And what that would mean if we had let that go without intervening is that you would only see WSB in that sort of middle region of chromosome 2 eventually. But what we decided, and I think many of our scientific colleagues decided, is that eliminating that region of meiotic drive on WSB, yes, we would lose one piece of founder contribution in that region, but at least the other 7 founders would have an opportunity to be of somewhat equal contribution within that region, and we wouldn't only have 100% WSB in that region. So that was a decision that we made several years ago to pursue eliminating that from our population. And it was sort of a big question, were we successful at that? We thought we were. We did all the genotyping to ensure that it was happening the way that we thought it would, but no one had ever checked on it. So this was really the first time that we looked to see to ensure that that region of meiotic drive was 100% gone. And that the other founders are sort of making up for loss in that region of WSB. And what you can see here in generation 34 is that once again, our sort of spark plots along the chromosome from each one of our founders look very similar to what was going on in generation 21. And we have confirmed that that region of meiotic drive on chromosome 2 is completely gone from our population and has not come back. The other great thing that we've noticed, since this was something that happened in the population long ago, that there was an allele from a specific founder that was going to fixation, we also wanted to check to make sure that that wasn't happening somewhere else without us knowing it. And by looking at all of this data, and we have lots more data to show that I can't show here in this presentation, but if you would like to contact us and would like to see more PDFs of this style data, we are more than welcome to share it. We intend on actually creating an annual genetic diversity report to report on the most recent generations of the JADO. to basically have transparency with our customers to ensure that we are breeding this population appropriately and that it is useful for their purposes in whatever scientific question that they have. So another thing that was going on in generation 21 and earlier generations of the JADO that we wanted to check in on was sex chromosome non-disjunctions. So there seemed to be in generation 21 a fair rate of XOs and actually XXYs, which we didn't look at here, but is reported on in a paper by Alyssa Chesler that reported on generation 21 and that WSB allele meiotic drive back several years ago. So for our customer satisfaction, we wanted to point out, sex chromosome non-disjunction rates in more recent generations. And what we're finding is that, even though there was a 2%-ish sex chromosome non-disjunction rate, I think they reported on higher rates in that paper that they published, but from the data that we had, it was fairly low rates. But in continuing in all these more recent generations, we've seen hardly any indications of sex chromosome non-disjunction in the data that we've had to work with in the populations we've had to work with. So that's really promising that things are going well. The other thing I wanted to show you is that throughout our generations, we're basically allowing random breeding to occur. We sort of control that randomness. But with every generation, there's going to be more and more recombination events that sort of split up where founder contributions are happening within the genome. So what I wanted to show you here is that basically the block size for how long of a tract there is of a certain founder before it switches to another founder is actually reducing with time. And this is something that's not unexpected. The more and more recombination events you have across the generation, the smaller and smaller those founder blocks are going to be. But what this actually does, and these numbers that you have here are in megabases. So in 21, it was like a 3.3 megabase block, and that's on average. There's obviously lots of larger block sizes within the genome as well. But the average of our recombination block size is decreasing. What that does is for people who are interested in actually mapping traits, associating the genomics with a phenotype of interest, their resolution for finding the allele or non-coding region that's actually responsible for their phenotype of interest is getting more and more narrow with time. So their resolution is actually going up, that they will be able to target the contributing regions more effectively and more quickly with less animals now that this recombination block size is reducing over time. The last thing about genomics that I wanted to point out is that our individual and population heterozygosity is high. So normally with inbred strains, you want all of your mice to be exactly the same, right? But this population, you want your mice to be different. The goal is to more closely mimic human populations and how different we are from each other. And with the goal of having each founder having 12.5% contribution to the genome, if you mathematically map it out, you essentially expect 87.5% heterozygosity within the population and hopefully within individual mice. What this shows is that at generation 21, we were pretty darn close to that. Our average was pretty close to an 87.5 average heterozygosity between individuals within a specific generation. That heterozygosity has gone down a little bit over time, but it's not anything very meaningful because our heterozygosity is still really high within individuals and within the population as a total. If you look at this y-axis here, we're talking 85% heterozygosity. So we've lost a little bit over the generations, which isn't unexpected. It's a closed population that we are trying to randomly breed within, but it's still very satisfactory, and I think should be received well by customers who are interested in genetic diversity, that the individual mice that you're receiving have lots of heterozygosity, and the population of mice that you're receiving have lots of differences between them. We also wanted to plot out our breeding statistics for this population as well. So what you can see here is that at generation 21, we had an average of about 7 to 8 pups per litter. Now, the number of pups per litter have increased over time, but once again, that's really to our benefit as breeders of this population, that we're getting closer to about 10 pups per litter on average. Obviously, there's lots of variation here. We have some litters with a lot of pups and some litters that are non-productive or just have 1 or 2 pups. So there's lots of variation here, and we've maintained that variation with a slight increase in the overall average per generation. But overall, I think we're maintaining our breeding stats very well over the course of randomly breeding this population in a laboratory setting. We try to be as unbiased as possible when we pick our breeders for the next generation. And I think this is showing that we're doing a good job at that and not doing artificial selection without sort of realizing it. Finally, wanted to show sex ratios. That's also a really important breeding feature for a lot of investigators who are using the JADO. Basically is because sometimes when you breed these different founders together, you can have skewed sex ratios on the basis of genetic incompatibilities and hybrid sterility. So to have litters that have even sex ratios is really ideal to show that the mixing of these inbred strains is going well. So once again, we've got lots of variation going on here. Our sex ratio is sort of looking at how many females there are. So throughout the generations, you've got some litters that are all female and some litters that are all male. But our average is really sticking at that 0.5 mark where we have even numbers of males and females all throughout the generations. We're really happy to see this data that our sex ratios are consistent and the sort of mixing of diverse alleles is going well and not affecting the sex of the pups coming out so much. It's not skewing to males or females in any direction. So with that, I'm just going to acknowledge the people that I worked on with this project, how he and Vivek Phillip from our computational science group really assisted with the genomic analysis and creating the pipeline to analyze all this JADO genomic data for this presentation and from here on out for our intended annual genetic diversity report on this population. I've got to thank our JAX Center for Systems Neurogenetics of Addiction. This is one of our NIH centers at the Jackson Laboratory, and they utilize the diversity outbred population a lot. All of this data came from them. They're really aiming for finding genetic contributors to different neuro diseases, and so especially addiction. And so they've been a heavy utilizer of the JADO, and they essentially donated all of this data to us. So if any of you are users of the JADO and are generating genomic data, we would be really interested to have you donate the data to us so that we can, for our more recent generations, generate these diversity reports more quickly and by having more data from more people. This is just data donation. We don't need to know your phenotypes or your QTLs that you're mapping for or anything like that. It's just, sort of giving back to the community to let us know that we're doing a good job at maintaining this strain and also showing the rest of the scientific community who's using this strain that it is still appropriate and relevant for their uses. So we think there's still a lot more time to go with this population. Our goal is longevity and maintaining this diversity for years to come.